Gene expression begins with transcription, the DNA being read to make the RNA working copy. And transcription is going to operate around something within the DNA called a gene, and we've talked about these before. Remember, a gene is a functional unit within the DNA. We're going to focus on structural genes. Structural genes are ones that will become an mRNA. So a structural gene in the DNA will be read to make an mRNA, or messenger RNA. The M stands for messenger. Other genes will become other RNA products. So other types of genes can become transfer RNA, which we'll talk about later on in this chapter, and ribosomal RNA, which we'll also talk about later on in this chapter. The majority of genes, over 90% of them, are structural genes. And again, that's what we're going to focus on. So each gene has three primary areas. They have, or rather four, I should say. So they have a promoter. A promoter is basically the beginning of the gene, and it's a specific sequence that marks it as the beginning. Next to the promoter, they have a regulatory sequence. A regulatory sequence is going to control how often that gene gets read. So that's what makes it regulatory. The third component is the transcribed region. This is the part that will actually become copied into RNA. So notice in the diagram, the promoter does not actually become part of the mRNA. It's outside of the transcribed region. So the copied RNA, the transcribed region, and again, this will actually become, or will be copied into RNA. And I'm running out of room here, so I'm going to go over to the other side. My fourth part of the gene is the terminator. And like that name suggests, it is the end of the gene. It marks the end of the gene, and also notice that it is not part of the transcribed region. So it's just an endpoint. It doesn't become part of the mRNA. So there are three major steps to transcription: initiation, elongation, and termination. And we'll go through each step here. So initiation, as the name implies, is how everything gets started. And so the first thing that has to happen is the recognition step. So this is the gene being recognized. In bacteria, which we're going to focus on, a protein called sigma factor recognizes the promoter. So sigma factor scans the DNA, and it will stop when it finds a promoter. So if you imagine your DNA as like, a pen or something, and my fingers are sigma factor, it's scanning along the DNA looking for the right sequence. And again, the right sequence is a promoter sequence. So it moves along and it scans. When it finds the promoter, it stops, and once it is attached to that promoter, it's going to recruit RNA polymerase. So sigma factor scans the DNA, stops when it finds a protein, and then it recruits to that location the other important enzyme in transcription, which is called RNA polymerase. So remember in chapter 11, DNA polymerase made DNA. So what do you think RNA polymerase does? Well, hopefully you can see that RNA polymerase makes RNA. So again, sigma factor will scan the DNA, it will recognize the promoter sequence, and then it will call over RNA polymerase, say, hey you, RNA polymerase, get over here, I found a promoter, and over comes RNA polymerase. Once RNA polymerase is at the promoter, it will start reading the DNA. So here's my DNA. So I'm just going to create a little bit of a sequence here. So there's my DNA. Okay, so RNA polymerase has been recruited over here. So just like in DNA replication, in order to get in there, RNA polymerase is going to have to peel these apart a little bit. Okay, but unlike with DNA replication, it's only going to read one strand. So RNA polymerase is only going to use one DNA strand. Okay, it's not going to copy both, it's only going to copy one. Just like DNA polymerase, it can only only make DNA in the 5' to 3' direction. So it can only make RNA in the 5' to 3' direction. So if it moves along the DNA moving to the right, and it's making it in the 5' to 3' direction, which strand is it going to use? So let me repeat this one more time because this is important. If I can only make DNA in the 5' to 3' direction, and RNA polymerase is moving along this DNA to the right, is it going to use the top strand or the bottom strand to make the RNA? Hopefully you can see that it has to use the bottom one. So notice, in order to make it 5' to 3' and be moving to the right, it uses the bottom strand. Whoops, and this is RNA, sorry, so we should be putting in a U. So remember, in RNA, I have to use a U, not a T. So U base pairs to A in RNA. So if I move to the right, I have to use the bottom strand, okay? If I had instead moved to the left, then I hope you can see that you would have to use the top strand. So again, moving to the left, I can make it 5' to 3', AGC, UA. And so the other thing I want you to notice is if I did this, this is the mRNA I make, 5' AGCUA. If I move to the right and use the bottom strand, then the RNA I make is 5' UAGCU. Are these two the same? So are these the same mRNA? And hopefully you can see that they are definitely not the same. So if I use the top strand, I make a very different RNA than if I use the bottom strand. So that means that both strands carry information. They make unique RNAs. And we'll see this again in a little bit. So notice that when I opened up my DNA here, it was a very small opening. So this is not called a bubble like we called it in DNA replication. And again, that's just because it's super small compared to what we see in DNA replication. So RNA polymerase is going to open it up a tiny, tiny bit, and then it's going to slide along, and the DNA will close back on itself, just how it was before. So remember that in transcription, the DNA goes back to exactly how it was before. That's the whole point. We're making a copy and leaving the DNA unaffected by this. So one term that I haven't mentioned yet that's on here is the template strand. So when I said I had to use the bottom strand, so here's my two strands of DNA, right? If I move to the right, I'm reading the bottom strand. So in that case, this is the template strand. So whichever strand is being read is the template strand of the DNA. So RNA polymerase is going to move all along the gene until eventually it reaches the terminator sequence. So remember the termination sequence, it's at the end of the gene. RNA polymerase starts at the promoter. So here's my gene. Oh, let me use something else. Here's my gene, right? I have, this is really confusing for me. I have the promoter at one end and the terminator at the other. So RNA polymerase is going to start at the promoter and work down and eventually come to the terminator. When it hits the terminator, it leaves and goes off of the gene, and that ends transcription. So let's look at the figure for this. So here's sigma factor. Sigma factor is recruited, or sorry, sigma factor finds the promoter and then it calls RNA polymerase over. So RNA polymerase comes and sits next to sigma factor. And then RNA polymerase is then able to start opening up the DNA. As it opens the DNA, it allows base pairs to form, only this time it's RNA. So if you have an A in the DNA template strand, so remember the strand that's being read is the template strand, and the yellow here is the RNA. If there's an A in the template strand of the DNA, then there must be a U in the RNA. If there's a T in the template strand of the DNA, there must be an A in the RNA. And this is happening in the 5' to 3' direction. So in this case, it's reading the top strand. It will move along the DNA, reading and copying the template strand. And then as it moves along, the DNA goes back to how it was before. So see how the DNA is rewrapped around itself. And the RNA molecule will be just coming off of there. So this is in the middle of elongation. The RNA is really long now. It's still reading the gene, and the DNA that's been already read is all closed back on itself. Eventually, RNA polymerase is going to keep moving this way, and it's going to bump into this Terminator sequence. When it hits this Terminator sequence, RNA polymerase is going to leave. So this is RNA polymerase falling off, and the RNA gets released, and the DNA is back to exactly how it was before transcription started. So this is what I was talking about earlier as well. So notice that this is a long piece of DNA. So this is a long piece of DNA with 3 different genes in it. And so in this gene, the promoter is found if I'm moving to the right. So remember, the promoter is a gene sequence, so it also has 5', 3' direction. So sigma factor is going to be moving along, and if it's moving along that direction, then it's going to find a promoter. If it finds a promoter moving this direction, then it will have to read the bottom strand. Because again, remember, it has to make the RNA 5' to 3'. So if the RNA is running from 5' here, it must be anti-parallel to the DNA, so it's reading the bottom strand. If I look at gene B here, I also find a promoter here while I'm moving to the right if I'm sigma factor. So that means I'm going to make my RNA moving to the right. This is the 5' end, which means I must also be using the bottom strand as my template strand. So in both these cases, the bottom strand is the template strand. Now if I look at this one, notice that the promoter is on the other side. So now I'm only going to see the promoter if I'm moving to the left. So my RNA is going to be made from 5' to 3' still, but I'm moving to the left. So now that means I have to use the top strand as the template strand. So now the top is the template. So either side of the double helix can be used to make an RNA, but it will make different RNAs and for different genes depending on where the promoter is. So practicing making sequences here, if a gene has the sequence 3' TAC GCC, what is the complementary mRNA sequence? Well, remember that RNA has to be anti-parallel to the DNA template too. It's going to be made 5' to 3', so moving to the right in this case. And then I make base pairs just like we did with DNA. So T pairs to A, and then A pairs to, and in RNA it's a U. So remember, this is one important difference between RNA and DNA. There's a U instead of a T. C pairs to G, G pairs to C, C to G, C to G. So the sequence here should be AUG, CGG. Okay, so I'm going to show you a movie here, so give me a second here. So again, as I was saying in class, it's important to watch animations. Oops, what's going on here? It's important to watch animations for this material. Oh, my mouse is disappearing. Okay, sorry about that. I don't know what's going on. I'll have to do the animation a different way, so we'll skip that for now. So I want to end by saying that we were focused on prokaryotic transcription, and eukaryotic transcription is really similar. Just in eukaryotes, we always have more proteins. So in eukaryotes, we actually have multiple types of RNA polymerase. RNA polymerase 2 is the main one that's used in eukaryotes. And also in that initiation step, in bacteria and other prokaryotes, we only have to use sigma factor. But in eukaryotes, it's actually going to require at least 5 different proteins getting onto the promoter in order to recruit RNA polymerase. So again, in bacteria, I just had to have sigma factor. In eukaryotes, I'll have to have at least 5 proteins all going on there at the same time. And that really allows eukaryotes to more fine-tune their transcription, rather than an on-off button, which is what sigma factor is. It's just one protein. Having multiple proteins the way that eukaryotes have allows it to be like a faster, slower thing rather than just an on-off thing. So a little bit more proteins, a little more fine-tuning in eukaryotes.